And I'm going to give you today a couple of stories. And I'm going to start, if I can get the right button, my disclosures. I'm going to start by talking about Coca-Cola. How many of you have heard about Coca-Cola? All of you. How many of you knew that actually Coca-Cola revolutionized the soda industry many years ago? So let me tell you the story. In 1930, Coca-Cola introduced the first soda dispensers. And the reason why these first dispensers were revolutionizing is because it was the first technology that enabled on-demand mixing of the carbonated water and the syrup, really changing that industry. These type of dispensers were located in many venues, including pharmacies where people were just getting around, drinking soda and socializing. Later on, in the 1960s, they launched the next generation of dispensers, the post-mix dispensers. This was an improvement over the prior dispenser, more compact with better mixing. And these type of dispensers were then started to be introduced in the fast food restaurants and cafeterias. But Coca-Cola was faced with a challenge that as they were expanding the menu of drinks that they wanted to offer to their customers, they were thinking, what is the next dispenser I'm going to need to be able to expand the menu of offerings I want to give? And with that, they launched the level-operated dispenser in the 80s. That was another revolution for the industry because in this case, now they were allowing, again, on-demand mixing of the syrup of the carbonated water, now with many different types of syrups, with a process that was reproducible in every single cafeteria and restaurant where they were using these dispensers. But the challenge didn't stop here. Coca-Cola kept expanding the menu of offerings, and they didn't have the right technology to be able to offer all these options to their customers. How were they going to now get this type of technology and add 30, 40, 50 type of levers to be able to offer everything they wanted to offer? They could not do that. So the big revolution actually came in 2009 when Coca-Cola launched the Freestyle dispenser machine. This was a touchscreen technology that allowed customers to be able to get more than 100 type of different flavors. This technology, this machine, was used in microdosing, and it was truly revolutionary. And what is kind of interesting about this Coca-Cola Freestyle machine is that this technology was developed in collaboration with Invitec, who was at that time a Danaher company. Coca-Cola launched, after many years, another free dispenser, the next generation, in 2018, that has more sophisticated touch screen. It also has an app. You can order your drink from your app. And they have continued to improve and change this industry. And you may be asking, what does this has to do with the genomic medicines space and rare disorders? I want to make a couple of analogies. First of all, this industry didn't have any need whatsoever to develop these technologies that could allow multiple processes and the on-demand manufacturing of multiple drinks until they had the challenge that they had to do it. So that was the moment that they innovated. Second of all, when Coca-Cola had this challenge on needing the right technology to be able to offer the variety of products that they had, they tried to get solutions that were making things simple. They were trying to reduce the complexity of this industry. And this complexity is a concept that is extremely important in what we are doing today and the conversation that we have been having for these past 2 days. Because if we look into what is the problem that we are trying to solve in the rare disease space, in the drug development for rare disorders, this is not just about a single problem. We are trying to solve actually an ecosystem challenge that we have. And this slide is not meant to be extremely exhaustive, rather than illustrating the challenge that we have in front of us. Let's start with the patients. I don't need to tell you this, but the patients are just waiting. They are waiting, and they don't have time. And the hospitals and the care providers are trying to take care of these patients, but they lack the tools for most of the time of early diagnosis. Now that's going to be changing with newborn sequencing. And they are trying to provide care, but they are operating in a healthcare system that is extremely expensive. And even if they are able to do this early diagnosis for the patients, they don't even know what to do. They have no ability to really connect the patients to therapeutic options, neither they know what to do with these patients. And hospitals and the patient community are looking into the developers and waiting for treatments. But in the for-profit industry, developers are always facing the challenge that they need to be thinking about the commercial viability of what they do. If you are a public company, you need to report on EPS. If you are a private biotech, you need to report to your investors. And even for those developers that want to help and provide some support, we know that they still face many challenges for patient identification, recruitment, and everything that, again, I don't need to explain to this audience. As developers are trying to advance the technologies that we have today for these patients, we then have an academic community that is continuously creating new information, an amount of information that sometimes is extremely challenging for us to really make something out of it and really digest what is that we can do with all this new information coming into the field. And then we also have the life science industry that is looking into this whole ecosystem, thinking, what other tools can I create to support you all? Because the life science industry has the tools that are used in hospitals to diagnose patients, to treat patients, by developers, by academic investigators. And they need to know what is the next tool that you need to be able to solve this problem. And this challenge about the tools that we need to develop medicines is now more important than ever, because we know that with genomic medicines, now we have the ability to really go after disease-causing mechanisms. We have the opportunity to really treat these patients. You have been talking about this over and over again for these 2 days. However, we need to be realistic. The curative potential of these genomic medicines, for rare disorders in particular, is not compatible with the current drug development model. Neither we have the technologies that will allow us to treat these patients, not yet, and it's just not compatible as well with our healthcare system. And what we need are alternative development frameworks that combine with advanced biomanufacturing innovation. We cannot do one without the other, to advance these treatments for the patients from discovery to the patient in a sustainable way. And I want just to spend 1 second about reflecting on this challenge on the biomanufacturing for genomic medicines for rare disorders. Let's not forget that in the world that we are living today, these genomic medicines are actually multi-component. This is no longer what we used to do many years ago. You just have one small molecule that you can formulate with many different excipients. The options were much more limited. Today we are facing multi-component therapeutics, and for manufacturers, this means additional challenges. You need to be able to do the right manufacturing for every single of the components that you have, and optimize not only the single components, but the final formulation. There are additional supply chain logistical challenges that we face in the industry. And of course, we are all waiting and looking for what is the right way for us to be able to do and develop these technologies. We need this regulatory guidance. So coming back to the analogy of the Coca-Cola machine, if we all want to try to decrease this complexity and continue to make significant impact in the space, maybe at some point we can all together collaborate and adapt this concept of the freestyle single process customization that Coca-Cola developed to do something similar for genomic medicines, where we truly have a process by which we can select the right gene with the right guide RNA, the right CRISPR enzyme, the right lipid nanoparticle, and be able to manufacture the right drug at the right volume, with the right quality, and the right time for the patient. So I think that this is really aspirational at this point, but I think that based on what we have been hearing for these 2 days, through collaboration, we might be able to get there. And this concept, this challenge, these problems are really important for us as the life science industry that we are. And I think at this point you've heard a little bit about Danaher in these 2 days, but again, we are a life science organization. We have a family of operating companies that have many tools and technologies that are being utilized in biotechnology, life sciences, and diagnostics. And we are here to help the whole ecosystem, trying to think, how can we do things differently? And the way that we are trying to help and to provide additional solutions in the space is by taking many of the technologies that we have across the portfolio and start providing integrated workflow solutions. How can we offer these less complex solutions for you to be able to advance in the genomic medicines space? And we recognize that we are not going to be doing this in isolation. Again, this talk is about collaboration. Trust me, I'm going to get there in a second. But at Danaher, we recognize that we are never going to be able to really make the right progress by working in silos. Collaboration is at the core of everything we do. It's because of that 3 years ago, we launched this Danaher Beacon program. This is mostly a program that is based on academic partnerships. We partnered with the best pioneers and innovators that share the same goal that we share in making impact in human health. We have beacons now in different domains, in genomic medicines, in diagnostics, next generation biomanufacturing, human systems, and of course data analytics with several institutions that you can see in here. In fact, we have a Danaher beacon with Oxford University with Julian Knight that is based on sepsis. But the beacon program I want to talk today is the same one that you heard from Fyodor on day one, is the IGI beacon collaboration. And what I want to do is to provide you just a different perspective from what you heard from Fyodor the other day. And I want to tell you a little bit more from the inside how this collaboration happened. Because again, we heard that it's not new about having academic industry partnerships. I think there is a different intent and flavor into what we are trying to do with IGI and Fyodor and everyone else's leadership. So everything started, and maybe Fyodor, not sure if you will remember this, at ISCT, in a coffee that you were sharing with Sadiq Kasim, that is our CTO for genomic medicines. And later, that was in May 2022, later that month, I met with Fyodor, was the first time I met him, and we continued the conversations, and we had an amazing conversation, I would say that was love at first sight, where we were finishing each other's sentences. And we were like listening to Fyodor, all the challenges that he faces when he's treating patients. And Sadiq and I are sitting there thinking, well, we might actually have the technologies that he needs to put them together and be able to get where he wants to get. So from all the many what if that we were discussing and debating that day, we decided to focus on solving one specific problem in this ecosystem challenge I mentioned before. That is the fact that the current system, and you heard this is actually from, still this one from Fyodor, you heard him yesterday that the way that they are approaching this N-of-1 development of CRISPR medicines for these patients, it is just not scalable. You cannot be deploying 4 years and $17 million every time you want to treat a patient. So what we decided was to join forces to try to get into building and actually going from this traditional, inefficient, and costly process to something that would be reproducible, standardized, and would be feasible to really scale up personalized CRISPR medicines for rare disorders. And as we were thinking about, okay, let's get it done, what do we need to do? There were 3 critical success factors for this to happen. Again, when you are trying to solve these challenging problems, you cannot just do one single thing. The 3 success factors were that we had to have the right development plan. The second one, we needed the right technologies. And the third one, we needed the right regulatory path. For trying to solve the right development plan and the right technologies, this is what with Fyodor and his amazing team and the Danaher team, we have been trying and working hard for more than a year now. I know it always looks so nice and seems simple when we are presenting these collaborations externally, but the reality is that there is a significant amount of work that goes behind the scenes to be able to put everything together. And here what we did was just, we are very thankful for our academic colleagues to embrace what we brought to the table, that was the rigor, the process thinking. Let's build step by step what this development plan is going to look like and put the right plan. This is not a PowerPoint exercise. We need to succeed in executing in this program. And this is just an example about one of those working sessions where we were working with the IGI team, bringing the Danaher business system to be able to map out every single step of what we wanted to do. And actually, we have Fyodor sitting in here doing his part of the contribution. So that was, having the right plan. Let's develop the right technologies. We are, of course, working on this. Hopefully, we will be able to provide at some point, maybe next year, more updates on this project. But importantly, what I want to mention in the last 2 minutes of my talk is that there is something else I mentioned as a critical success factor, that is a regulatory pathway. And you've heard that throughout the 2 days here, that the FDA issued this Platform Designation Act. So for us, this was like all the stars aligning. This platform designation is aiming at providing guidance into how can we platform the different components of these multimodality treatments that I mentioned before. However, this this guidance still is missing the concreteness into what exactly does it mean. So what we need to do now as a next step is provide the guidance and start thinking together with the regulators, what does it mean to platform indications? How do we need to build this type of guidance? So what we are doing right now is working together with the Alliance for Regenerative Medicine, ISCT, to put together a scientific exchange meeting. This is going to happen in November. This is going to be, live stream. Everybody can see and attend virtually in this, scientific exchange industry roundtable, where we are going to be sitting with the FDA, providing clear examples and suggestions into how do we need to be building the thinking around platforming gene editing. And I want to spend a second into what is the objective for this particular scientific exchange. As you can see, we are very focused in trying to provide guidance for scientifically feasible but commercially non-viable approaches. Importantly, what we want to do is to identify a regulatory path or provide recommendations that can allow us to have this fastest time to patient. This is extremely important for us. And for this, I know that we don't have too much time, but I think at this moment, I think it's important to recognize that we have been talking about platform designations and how this could significantly and materially change the industry. But again, we are really missing at this point the concreteness of what we need to do. The most important, I would say, from all the promises of all these platforms is going to be not just the speed. Hopefully, it's going to be reducing the cost of medicines. This is a topic as well that we've been hearing over and over in the organization. So this is an aspect that we want to cover as deep as we can in this exchange. And I know I'm not going to have time, but really the thinking is going to be also important. We have a guidance, but we don't have a solution. And when it comes into thinking on the different aspects of platforming, a therapy, what does it mean? There are components on the platform technology that are going to be based on the therapeutic target. These are the variables that we can think about. It's going to be the disease, the indication, the organ. There are as well aspects of the platform that are going to be related to the delivery method, the gene editing method, the editor, the guide RNA. Of course, there is going to be a significant component of this platforming that is going to be related with biomanufacturing and the quality control and the CQAs that we need to put in place to ensure that while we are trying to streamline a process, we are not losing the rigor and the quality that these patients deserve. And then the route of administration and considerations related with the clinical trial. Depending on the technology and the indication, some of these are going to be fixed, some of these are going to be variable. This is where we need to put the thinking to provide the recommendations for how to platform indications, how to platform therapeutics. And that's everything I wanted to share today, and I want to go back to what we told with Fyodor many times. Fyodor, if we can dream it, maybe we can do it. So let's keep working together to try to make impact in this space. Thank you, everyone.